Question number 1. Enumerate the modes of genes action on determine the phenotypes and genotypes of the progenies produced of each modes. There's are 4 modes of genes action including additives, complete dominance, epistatic, and incomplete dominance. Additives. Both alleles equally and unidirectionally contribute to the generalization of the trait when neither is dominant, and between the two homogeneous phenotypes, the heterozygous phenotypes is in the middle. Epistatic interaction. The generalization of a phenotype is controlled by epistatic gene interaction when particular arrangement of alleles from various genes are required. Epistasis is the process which alleles from 2 or more loci interact to create a phenotypes that neither gene alone would be able to create. Complete dominant. There are only 2 phenotypes when full dominance is a manner of gene activity, and 3 phenotypes because in the heterogeneous two different types of alleles for single traits condition. The expression of the recessive alleles is hidden by the presence of the dominant phenotypes. Incomplete dominant. When the dominant alleles manifest itself, more dominance take on another form is stronger than recessive alleles, but not efficiently strong to match the homogeneous dominant phenotypes in the heterogeneous state. We refer to this kind of dominance is incomplete dominance. 3 genotypes and 3 phenotypes are produced by genes with this method of action. Number 2. Different dominant from recessive genes. Dominant is always expressed when present. Recessive is only expressed when no dominant genes are present. Number 3. A dominant G alleles common CARP produce a normal pigmentation of a recessive alleles produce a gold pigmentation. A cross between normally pigmented gold common CARP produce all normal progenies. Letter A is what are the genotypes of the parent? The genotypes of the parent is a big GG normal pigmented, and big G and little g is a pasanemic pigmented. Little gg is golden. Letter B is what are the genotypes of progenies? The female gametes is a big GG and the male gamete is a big GG and the female gametes is a small GG. Only what could be for the simplest of Punnett square will cross 2 genotypes, same genes, but may or not contain different alleles. Look at the example of a genetic cross for normal pigmentation and gold common CARP. The gold common CARP is being dominant and the normal pigmentation is being recessive. And we use a big G, little g for the male gametes and the little g for the female gametes. This cross will draw a square split into 4 quarters, like 4 panel window. This is a Punnett square on the parental genotypes is will go on top, on other will go side. Inside it doesn't matter which genotypes goes where, but were conventionally write the dominant alleles. Two possible genotypes, big G little g and little g. Its genotypes has a 50% chance of showing up spring. Similarly, it have a two possible phenotypes, gold common carp and normal pigmentation, with a 50% of each. It is a possible 50% produces all normal progenies. Letter C. What genotype should be mated to produce on old gold phenotypes? Little gg genotypes to produce old gold pigmentation. Number 4. Based on the theory, discuss the hybrid cross. A is list of gametes produced by a hybrid cross of two homozygous gray guppies with curved spine. A transparent female guppies can be produced if the male is transparent and the female has at least X allele, and the transparent tail male guppies are produced only when the female has the recessive X allele. Letter B. Draw the Punnett square and determine the genotypic and phenotypic ratio are expected from the cross. The male gametes are X, Y, and Y. The female gametes naman is X and X, A, X, A. The crossing of female gametes and male gametes is XX transparent paled male and XY transparent tail, and the crossing of male gametes and female gametes is XX caudalist female and XY caudalist female. Number 5. Based on the given example on X-linked genes, how are transparent tail guppies produced? And prove your answer. Then draw a Punnett square and determine the phenotypic and genotypic ratio of the progenies of the cross between the mating and homogeneous caudalist female and a transparent tail female. Draw a Punnett square and determine the phenotypes and genotypic ratio of the progenies of cross between the mating of heterogeneous caudalist female and a transparent tail male. XX times XY. The XX is a caudal female, XY is transparent tailed male. Male gametes and female gametes, crossing of a caudalist female and a caudalist male. Then the XX is crossing 2XY. That XX is transparent tail female and the XY is a transparent tail male. Genotypes ratio is 1XX and 1XY. 1XX is a 1XY. Phenotypes ratio is a caudalist female and one caudalist male, then one transparent tail female and the transparent male. Number 6. Determine the factor that causes variations among organisms. It is a mutation that changes in a sequences of genes in DNA are one source of genetics variation, another source of genes flow, or the movement of genes between different groups of organisms. Finally, genetic variation can be result of sexual reproduction, which leads to the creation of new combination of genes. Number 8. What is a genetic drift and how it can be prevented? Genetic drift is a random change of allele frequencies within a population. It is one of the main mechanisms that drive evolution. The regular replacement of foundation animals from genetically fixed embryos is the only way of effectively slow down genetic drift and prevent genetic changes from accumulating. That's all. Thank you.